Hello, good morning. Myself Shilpa Patil. Today we are discussing about the non-cyclic photophosphorylation. First off, we are saying the non-cyclic. There is the two types of phosphorylation which takes place in the plant for the process of the photosynthesis. The non-cyclic photophosphorylation and cyclic photophosphorylation, which is take place in the chloroplast. Okay, let's see. We are saying the first of the location of this reaction. So this is the chloroplast, okay, and in the chloroplast we are seeing the disc-like structure is known as a thylakoid, and the our pigment system, okay. So the pigment system is situated on the membrane of the thylakoid. Where is the pigment system is situated? Pigment system is situated on the membrane of the thylakoid. So this reaction we are see here. The all the pigments are situated in the thylakoid. There are the different kinds of the pigments which are help in the process of the photosynthesis. So then this place is known as a lumen, which is present in between the thylakoid, and this is the stroma. Okay, this stroma is also help us for the whole process. So let's see our reaction. This is the non-cyclic photophosphorylation, which takes place on the thylakoid membrane. So first of the first event of our reaction is takes place and start from the water splitting. Water which is absorbed by the root, okay, which is transferred toward the our chloroplast. When there is the photon of the energy which is strike on the pigment system or in the chloroplast, then there is the first event which is takes place is the photolysis of water. Photolysis. Photo means sunlight. Lysis means breakdown. In the presence of the sunlight, there is the breakdown of the water molecule. When the breakdown of the water molecule, we are found there is a formation of the four proton. When the proton is released, with the proton there is a release of the electron also, and there is a release of the O2. Okay, this is the O2 which is formed in the process of the photosynthesis, okay, and which is a release of the through the stomata. It's a first product. Then when the proton is formed, proton is uh, placed in also in the stroma, okay. So due to the proton there is a formation of the proton gradient, and this proton gradient we will see this is also we are help in the process of the photosynthesis. And when the electron is released, this electron is transferred to the pigment system. Second, okay, here on the thylakoid membrane, a pigment system. Second, it's activated on the 680 nanometer sunlight, okay, wavelength of the light. When it got the 680 nanometer of light, the electron get excited, okay. When it's the free electron, when the free electron get energy, it's also going in the excited state, okay. Highly energetic electron. When the highly energetic electron is at the second. PS second is goes into the excited state, and then the PS second release their electron to the next pigment, okay, which is also switched. All these members are situated in the pigment thylakoid membrane. This electron is transferred to the our first pigment is the plastoquinone. So electron is goes to the plastoquinone. Plastoquinone also release this electron because this electron has a very higher energy, okay. And if the any pigment system absorb this highly excited energy, then this molecule will be break by this highly energetic electron. Hence plastoquinone gives this electron to the cytochrome B6. When the electron is transferred from plastoquinone to cytochrome B6, due to the pumping of the electron, there is the formation of the 1 ATP. Okay, so this ADP and IP is combined in the presence due to the transfer of the plastoquinone to cytochrome B6. There is the formation of the ATP. Okay, this is our the first product of this reaction, which is the first ATP. First ATP is formed. Okay, so we are also got the question from ATP is formed where location of the ATP's formation is the when transferring from the plastoquinone to the cytochrome, there is a formation of the 1 ATP. Okay, this is the first event. Okay, here we can be got a question. So then the cytochrome B6 give this electron to the plastocyanine and plastocyanin give this electron to the pigment system first. When the electron coming from this rotation, so the electron lost their energy. When the electron goes to the pigment system first, okay, which is activated at the 700 nanometer, it's the pigment system say first is also get the photon of the light. Photon means quantum of the energy. Photon of the light. This electron is also goes in the excited state. When the pigment system is excited, the pigment system gives this electron to the peredoxin. Peredoxin is the first only one acceptor which can accept the electron. Okay, so this electron, when the peredoxin accept this electron, peredoxin NADP reductase is gives this electron to the NADP positive. NADP positive, when the one electron is got, then NADP positive, then there is the bind of the NADP H proton. Okay, one electron is bind with the one proton, so there is the formation of the NADPH. Okay, this is our the key product of this reaction, which is the NADPH. Okay, so what we got from this reactions, one ATP and one NADPH. Okay, so what is the specialty of the peredoxin? Peredoxin is the strong acceptor, of this reaction. Hence, this is one of the reason that the non-cyclic reaction it takes place due to the presence of the peredoxin, because peredoxin is the strong reducing agent which has capacity to accept the electron. So what we are discussing here, so the electron is transferred from the pigment system second to the pigment system first, and pigment system first give this electron to the peredoxin. In this event, what we are seeing, the most important thing in this reaction, there is a involvement of the both pigment system first and pigment system second. Okay, all these are the questions. यहाँ से question बनेगा. So the pigment system first and pigment system 2 both are involved. They are activated on the 700 as well as the 680 nanometer linear electron flow. Okay, this reaction is also known as a linear electron flow because these are situated on the thylakoid membrane. Then another one event is most of important is the photolysis. Okay, so the photolysis takes place here. Then we are getting these products. Which are the products we are getting here? We are getting the one ATP, one NADPH, and oxygen. Okay, all these products are released in this reaction. Then what is the final electron acceptor? यहाँ से भी question बनेगा. So the most final electron acceptor is the peredoxin. So I hope so you will be must be understand this. Okay, thank you.